Hello, everyone. Thank you for coming to the next talk in my Science and Religion series. Just a reminder, if this is your first time, my name is Dr. Christina Sweeney Hillesheim, and this is just one talk in a series of talks that are focused on different topics related to science and religion, specifically Christianity. If you'd like to know more about me, you could go to the first talk. I do a little intro, more about my educational background and where I'm at spiritually as well. But so happy to have you. Just to let you know, this is going to be the first of two talks. This is like a part one and a part two. Just because it's longer, I don't want you to have a really long video. All right, so the challenge of DNA. So we're going to look at DNA in relation to evolution. So just to give you some facts about DNA, since I'm not sure exactly where you're at in your science background. So DNA stands for deoxyribonucleic acid, and it was identified in 1953 by Francis Crick and James Watson. There's actually a pretty interesting story about them finding it and going into a pub and going like, "We found it! We made a great discovery!" So it's kind of exciting. But apparently, Crick's wife was like, okay, sure, you say it every day. But they finally actually did have a great discovery. So, what was discovered is that the molecular structure is a double helix. So, kind of this double spiral that you can kind of see in the background picture here. It's actually 6 feet long and tightly coiled inside every one of our body's 100 trillion cells. So, we have a lot of different DNA molecules. It uses a four-letter chemical alphabet that I will talk about in more detail on the next slide. And the important thing about DNA is it stores the language of life. So it stores information that we need for life. And it is the precursor, it's what we need. This information is how we create all the proteins. And proteins are, you have to have proteins or we can't have life. So proteins are kind of like the basic building blocks, and DNA is that information, that code that helps us make those proteins. Each one of the 30,000 genes that are embedded in our 23 pairs of chromosomes can yield as many as 20,500 different kinds of proteins. So, with this information that we have, it's not just like, oh, here's one recipe, it makes just one type of cake. We actually can make a lot of different types of cake from this piece of information. So, this information is very broad, if that makes sense. Lots of options based on this four chemical alphabet. So, just to give you a background, so here's on the right here is this double helix. And is the four-letter code alphabet that I was talking about. So DNA's instructions for building a protein have four-letter code. And this four-letter code represents four bases. And so these bases, A, T, C, and G, are adenine, cytosine, guanine, and thymine. And they always pair up together. So A is always paired to T and G is always paired to C. So if you look in this picture here, you don't see A ever with G or C here. It's A with T and G with C. So there is sort of like a code or a structure of how it has to go. Now, so arranging these four bases will instruct the cell to build different sequences. So here we have like the T, then the G, but it could be T, then T, or T, then C, or T and an A here. So that's where we get that variation. But if there's a T, then on the other side, it has to be an A. So that's where we see that kind of instruction is because there is strict code or strict rules on what can be attached to what. So these four bases instruct the cell to build different sequences of amino acids. And these amino acids are the building blocks of proteins. So different bases give us different amino acid sequences. So depending on the order of these bases, we get different amino acids. Now, then what is an amino acid? So amino acid is a part of a long chain that makes up a protein. So you could kind of think of protein as like a train or a long chain. So protein is along kind of like a chain, a train, and each kind of car on the train is like an amino acid. Is how you could think about it. And so there's a long array of these amino acids. that make up the protein. Now, each protein is going to be different. It's going to be bent and twisted into a three-dimensional shape. And that's based on this amino acid sequence, how it's going to be able to bend. And the shape is really important because the shape is kind of like a key. So the way that some of these proteins work is it's kind of like a key and a lock. And so the shape is like the teeth of the key that's going to be for a specific lock. So the shape is very important. It's not just an arbitrary aspect of the protein. Not only does it determine, how it fits in lock, key to lock, but it determines the function of the protein, how it's going to work. So if it's unfolded, it's not going to function like if it were folded. And if it's unfolded, it won't work at all. So that shape is very important for proteins, what their purpose is and how they'll function. And they're made out of the bases, then the amino acids. So just to kind of give you a little bit of a background of how this works in relation to, I talked about proteins and DNA, but how does this come together? So there's a process. So we have the DNA right here. Now we can replicate the DNA, but that's not what we're worried about right now. We're worried about trying to make a protein. And so the first step with DNA to make a protein is transcription. So what happens is we have a molecular machine, and we had a whole lecture on these molecular machines last lecture. So you're welcome to kind of go back. But these are some examples, more examples of molecular machines that I kind of talked about before. So here, a molecular machine is going to unwind a section of the DNA. So here you can see the DNA is all wind together. And so it's going to come and it's going to kind of separate that winding. So now you have that separation. So molecular machine unwinds a section of the DNA helix to expose the genetic instructions. So now we can assemble. So remember, we have those bases that were paired together. So now they'll be separate. Now the bases are separate. And so now we can kind of match that code, that pattern. Then another machine is going to come in and copy these instructions, copy that code, and that's the messenger RNA. So the messenger RNA comes in and matches it up. It says, okay, you've got an A, I need a T, you've got a C, I need a G. You kind of match it all up. And then you have this slender RNA that is carrying that information. It's going to carry that information out of the cell nucleus and into our ribosome. And remember, we also talked about the ribosome before. It's this two-part molecular factory, and we talked about all the things that kind of happen in this molecular transport factory system. So now that the ribosome has the messenger RNA, and now we're going to have a molecular assembly line that's going to come and build the specific chain of amino acids. So now they're going to come in and build those amino acids. Those amino acids are going to be transported from other parts of the cell. They're going to link into chains. And the chain here, so think about like a train that has like a few cars. These are like about 100 or more. So a lot of these little cars in this long chain. And then, and remember, it's really important. If this car is in front of this car, if this amino acid is here versus here, it really changes the type of protein we're going to build and the function of that protein. If it is even going to be functional, because if we move this one, now it might not bend and be able to have the structure it needs and then have function at all, or it might have a different function. So that determination of these amino acids, that order in the chain is really important. So then once the chain is there, the chain moves from the ribosomes to a barrel-shaped machine that helps fold it into the precise shape. So then it looks at, okay, we've got these amino acids here. It looks at the order and it starts to kind of fold it and bend it into the correct shape. And then another molecular machine is going to transport that folded chain, that folded protein, to exactly where it needs to be to kind of be able to do what its function is. So DNA, RNA, and then we have our protein. It's kind of the process here. So that's how DNA relates to how do we get proteins. Now, I want you to think about DNA is like a library. Because remember, I said it's not just one piece of information. DNA has a lot of information. So when it comes in, when an organism comes in to access that information, think about it like a whole library. When it comes in, it's picking a specific book that it needs from the DNA to build its critical components. So the part of the DNA that's open, that it's reading that code. So it's not just, oh, come in and get one recipe. You have a whole library. You have a lot of information. So remember in DNA, there's long lines of A, C, G, and T's precisely arranged in order to create that protein and folding structure and folding. Now you might think, okay, so I know I have one protein. It needs at least 100 amino acids. And so then, but how many bases do I need in each amino acid? Like, what are we thinking? So one protein needs about 1,200 to 2,000 letters or these bases. So it's a lot of detail. So we're not talking about a little bit of information. It's a lot of pieces of information that is provided here. But the big question in this presentation that we're trying to figure out is, where does that information come from? Okay, so this is very similar to the previous talks that we had about information. So where is that information coming that is now being used to create those amino acids and then to create those proteins? That's the big question. So how can we answer this? So you might ask yourself, well, how can I look back in history, thinking maybe about evolution or the beginning of the universe, beginning of life, and go, well, that was way back in the day. How can I even make a prediction of how that happened and make a reasonable prediction? So the reason that we can do this is based on an idea called uniformitarianism. And this is the idea that our present knowledge of cause and effect relationships should guide our construction of what caused something to arise in the past. So, how we understand how things work now can help us make guesses on how things happened previously. And you do this every day, you don't even realize it, but I'll give you an example. So, say you find you're walking around maybe in like the desert or, in some woods, and you find these like ripple marks on this really old piece of sedimentary strata. And you're like, hey, I wonder where those ripple marks came from, like what caused them. Well, in present day, you see these same sort of ripple marks are being formed in lake beds. So as the water evaporates, it causes this ripple mark. So then you could say, oh, okay, so one of the causes of these ripple marks could be from this evaporation process. It could be similar to what caused those ripple marks in that really old sedimentary strata. So this is how you can make kind of a hypothesis of what you think, what you see now as cause and effect as to what could be the answer previously. So even the very simplest cell, we're talking about today's simple cell or in the fossil record, requires information. So it's not something new that, oh, now we needed information. It's always needed information that was stored in the DNA. And so that information has to come from somewhere. And Henry Quasler stated in the 1960s that the creation of new information, so if we're talking about brand new information, is habitually associated with conscious activity. So I'll give you kind of an example of how we think about that. So think about, say, maybe DNA is like a software program. So there's code, and using that code, I can develop something. But how did we get the information for that program? Those programs didn't just appear. Those programs required an intelligent programmer to develop them. And from that program, now you can get other things created from it, but something had to put in the information for that. So if we wanted to use uniformitarian logic, if right now we have information and we say, oh, new information was created, it must have been created from some conscious activity, from something thinking about it, then that would be the same for in the past. So the cause of ancient information, like in the first cell, must also be the product of intelligence. And so the information needed for life was created by an intelligent designer. Like the information needed in a software program was invented or created by an intelligent programmer. So, but let's look at it from what evolution has said. What did the father of evolution, Charles Darwin, what did he say? And you may have heard kind of the prebiotic soup, and you may have heard something mentioned about this. So let's just talk about this. So Charles Darwin wrote a letter in 1871 that kind of just talked about what he thought was this process. So steps of the theory that he shared is as follows. First, we need a buildup. So we need a buildup, and the buildup would occur. How this is happening is by pure chemical synthetic processes occurring on the surface of the early Earth. And we need a buildup of all the basic organic compounds necessary for the formation of a living cell. So we need all of kind of the puzzle pieces to kind of exist, and we need a lot of them. And Charles Darwin believed that they would accumulate in the primeval oceans, kind of creating this nutrient broth. And that's where we get that prebiotic soup that comes from, is that all of these little pieces are mixed together in this ocean. And then in certain specialized environments, meaning, the right temperature, right conditions, pressure, etc., these organic compounds were assembled into large macromolecules. So now you've got these little bits and pieces, kind of, you can almost think about them like Lego pieces, and now they're kind of coming together organically in this big soup to form bigger pieces, macromolecules, such as proteins and nucleic acids. So think DNA, RNA. And then over millions of years, over time, these combinations of these macromolecules occurred, and which were then endowed with the property of self-reproduction. And so now DNA can replicate and have more DNA, and then other things can do this as well. And this is all driven by natural selection, where then natural selection is going to make it even more efficient. And then because it's more efficient, we'll have complex and more complex self-reproducing molecular systems that are going to evolve until we finally have the first simple cell system. So this is how Charles Darwin kind of explained this whole process, from having nothing to creating some of these compounds all the way to having our first simple cell. So, how do we kind of think through this? What is the scientific evidence for this? So, let's talk about this evidence. There actually isn't any evidence for this prebiotic soup that was Darwin described. And remember, Darwin described this a very long time ago, before there was a lot of advancement in science and technology. So, he was just making a guess based on what they knew at that time. If this prebiotic soup existed, it would have been rich in amino acids. So there would have been a lot of amino acids in this soup. Because remember, amino acids are the building blocks of proteins, and we have to have proteins to have life. And there would have been a lot of nitrogen, because in amino acids, there is a lot of nitrogen. They're very nitrogenous. So if there would have been a lot of nitrogen in that environment, then we would have seen remnants of this nitrogen in the earliest sediments. So we can go through, we can look at the minerals at the rock of the older part of the earth, and we should see this nitrogen-rich deposits. However, after lots of looking and exploration, these deposits have never been found. And Jim Brooks wrote in 1985 that the parts that were ancient, that were very old sediments of the earth, where it should have high nitrogen content, is actually really low. It's relatively low, just 0.015%. So it's not like, oh, it's kind of nitrogenous, but not as much as we thought. It's not even close. It's not even like, oh, we could kind of maybe argue for it. It's very low. Also, additionally, one thing that is not mentioned is if we have all these different chemicals together, you're going to end up having problems with cross reactions. So you want two things to react together, But then you have these other things that are going to come in and try to also react. So cross reactions are reactions that we don't necessarily want to have happen. And so the amino acids are going to end up reacting with other chemicals. And this will actually make a sludge that's not really life friendly. And this was actually done in a famous experiment where they were trying to replicate what this prebiotic soup was like, tried to put in different chemicals, amino acids into it to see what would happen. And it literally just came out with this sludge because of these cross reactions. And so the few experiments that have been done to kind of provide some scientific evidence for this prebiotic soup has just shown that it doesn't work. It just kind of creates a big, ugly mess that is not friendly to life and doesn't get you anywhere. Okay, so what are some potential naturalistic routes to explain where we're getting this information? Where is this information coming from in DNA that maybe it's not coming from the prebiotic soup, or what's another explanation? So there are three possible explanations that I'm going to discuss: random chance, natural selection, and chemical affinities and self-ordering. But since this talk would be really long if I did all three, for just this part of the talk, I'll do one and two. And in the next part of the talk, I will talk about three, the chemical affinities and self-ordering. And then I'll talk about a little bit more about evolution and what that means in the fossil record as well. So you can look forward to that talk, or you can go ahead and watch it on YouTube now. Okay, so let's talk about random chance, scenario one. So it's interesting because this one is presented regularly, but it's not presented in all different groups of people. So if you were to talk to a group of scientists, they would not actually use random chance as an argument. Even though if you were to read a popular journal, or if you were just to talk to some people on the street, then random chance is actually believed by the general public, but not by people who have really studied this. And the reason that it's not really believed by people who have studied it, either scientists or mathematicians, or even the general public that have just spent time to really look into this question, is due to the unrealistic probability of this occurring. And so for there to be a protein, the simplest protein, there's a minimal complexity threshold. So we talked about the protein having those amino acids, but how long does it have to be? How many of these little cars do I have to have? What's the simplest protein I can make? And so to have a simple protein, you have to have that 3D structure. So just having the chain doesn't cut it. You have to have the chain, and then it has to fold. And when it folds, that's called the tertiary structure. And so the tertiary structure is the minimum level of folding a protein has to have to perform a function. And so the simplest protein that we need, it still needs that folding structure. So it needs the alpha helixes, the beta sheets, which you can kind of see in this picture, and it needs at least 75 amino acids. So just to kind of give you an idea of how probable it would be for a random chance to create even the simplest version. So let's kind of think through it from the math. So what do we need to form a protein molecule by chance? So first we need the right bonds between the amino acids. So we have this amino acid kind of train car, and then the next one comes. But we need the right bonds between them to kind of attach all the amino acids in the chain. Also, you need the correct amino acids. So amino acids come in right-handed and left-handed versions. So it's kind of the way that they're spiraling in the direction they have. And so, and this is true with other chemical compounds. It's called stereochemistry. So this is pretty common concept in organic chemistry, but we're discussing it here in amino acids. In some compounds, the right and the left hand are both beneficial, or in some, for example, headache medicine, one is really good and one is not good. So sometimes one functions and one doesn't function, or they have very different functions, depending on which molecule or amino acid we're talking about. But in this case, we need amino acids that are only left-handed versions. So we would need only the left-handed versions to kind of come into our chain, and they need to come up in that specific sequence, like letters in a sentence. So if I am trying to write the word probability, but I start with a B instead of a P, then I'm already, from the beginning, not doing it correctly. So we need the amino acids, we need the correct one, and we need them in the right order. The probability of this happening on its own is 10 with 125 zeros. So it's 1 to the 10 trillion, 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 trillion. Okay, so 125 zeros. Now, that's only one protein. That's just to make only one protein. We actually need 300 to 500 proteins in a simple cell. So we can have one protein, we can spend all this time and all this effort trying to get one, and we're still not going to get anywhere with life. We need much more. And this is all accomplished in only 100 million years, because that's when we start seeing evidence in the fossil record. So that's when we know, okay, if this were going to happen, it has to have happened by this point. So in 100 million years, we need to have 300 to 500 proteins, and random chance just runs out of time. It's just not possible probability wise for it to occur. So that's why random chance is not really is not accepted in the scientific community. So now let's talk about the second one. The second one is natural selection. So let's talk about natural selection. So natural selection doesn't actually work at the level of chemical evolution. And let's explain why. So we have to remember, what is actually natural selection? What is the theory of natural selection say? So first, for natural selection to occur, you have to have an organism that reproduces. So this organism reproduces, it has offspring. And the offspring aren't the same. Similar, but not the same. And whichever one is better adapted to the environment survives. And then these adaptations, because this one reproduces, then moves on to the next generation and so forth. But for this to work, natural selection has to start with a self-replicating organism. Natural selection cannot explain how self-replication starts. It needs that as the beginning step. So without self-replication, natural selection can't explain it. It needs that to start the process, because the organism has to reproduce, and then theory of natural selection can explain. So when we're talking about at the chemical level, to have reproduction, we need cell division. But for cell division to occur, we need the information that's rich in DNA and proteins. And so here's the problem, is we need the information in DNA, we need proteins, then we can get cells, then we can get life that's reproducing. But natural selection is trying to explain all of this all the way to the beginning, but it's assuming that we have all of this to begin with. Okay? So it's kind of trying to have a circular argument, because DNA and proteins are the very things we're trying to explain in this talk. And natural selection is saying, well, I need those things to even do my business, pretty much. So some people say, okay, fine. We're going to say that it wasn't DNA, but it was RNA. So it's the RNA first hypothesis. And this became pretty popular at one point. And so what it says is, what if the reproductive life originated in RNA, and then RNA was used then to build up to DNA? So we started with RNA and then to DNA. But there's still a lot of problems with this hypothesis, because the RNA molecule also needs information to function. If you see in this picture here on the right, the RNA looks very similar to DNA. They're both nucleic acids. They both have this sugar phosphate backbone. They still have these bases, these nucleobases. So it's very, very similar. The difference is what those bases are exactly. but it still is rich with information. So the same problem that we had with DNA, where does the information in DNA come from? We have the exact same problem in RNA. Where does that information come from? So for a single strand of RNA to replicate, we actually need another RNA. So for this RNA to replicate, we need another RNA close by. And if we're talking about this big soupy pool, what is the probability of these two identical RNAs, not similar, identical RNAs being next to each other? And this was actually determined mathematically. The probability of having these two molecules close by is 10 billion, 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 billion. And yes, I meant to write 6 billions there. So not 6 billion, but billion times billion. So 6 billion 6 times. Okay, that is how unlikely it is that these two RNA molecules, one would even exist, but then they would be next to each other. And it's also not probabilistic. So the RNA molecule, the RNA first hypothesis really doesn't even still answer the question, because the question still is saying, where does this information in RNA get? So natural selection can't answer the question because it needs the answer to the question for it to even continue to describe things. So in my next talk, in part 2 of this, we're going to go through what is argument number 3, which is the more stronger argument of the 3. You can kind of see that they build up in strength. And then we'll talk about, well, what does this mean and how do we then think through evolution and the Cambrian explosion and the fossil record. So I hope that you will continue and watch my part 2 of this talk. Thank you for coming to this talk. Here are a few references, including the reference for the letter that Charles Darwin wrote with that theory. And if you want to dive deeper into some of the probabilistic calculations and explanations, you can look at those here. Thank you so much.